Hi, my name is Marco Ferreira. I'm a professor of statistics in the Department of Statistics at Virginia Tech. Today I'm going to talk about BG2, Bayesian verbal selection in generalized linear mixed models with non-local priors for non-Gaussian GWAS data. This is joint work with Xuan Xu and Jake Williams. If you have any questions, you can send me an email at marf@vt.edu. Here are some acknowledgements. This research was funded by grants from NSF. So grants NSF DMS 1853549 and NSF DMS 2054173. Computations for this research were performed in supercomputers of advanced research computing at Virginia Tech. This talk is based on a paper that came out in BMC Bioinformatics this year, 2023. And if you want to use our method BG2 to analyze GWAS data, then there is another package available on Bioconductor. The package is called BG2 as well. And here's the address and the DOI. Here's an outline of the talk. I am going to start with motivation. After that, I'm going to talk about generalized linear mixed models for GWAS. And then I'm going to move on to the integrated likelihood, non-local prior. After that, I'm going to talk about how we perform SNP search. And then I'm going to talk about an approximation we use, that's population parameters previously determined. After that, I'm going to give some details about screening and model selection in our method. Then I'm going to present some simulation studies, some applications, and then I'll provide some conclusions. Motivation. So our objective here is to find which genetic variations in the DNA are related to observed phenotypes. Here is an example on the right where we have 4 different ecotypes of a same species, and we have 2 treatments: control, where the plants are watered with just normal water, and treatment, where the plants are watered with salty water. And what happens is that the plants that are watered with salty water are not as healthy as the plants that are watered with usual water. But there is some difference in how different ecotypes deal with the salt. So some ecotypes are more resistant to salt than other ecotypes. And then one of the objectives here would be to identify which ecotypes or which genetic variations provide this better ability to deal with salt. So again, scientific question: which genetic variations influence or predict observed phenotypes? And in this talk, we're going to focus on a particular type of genetic variation, which is called single nucleotide polymorphism, or SNP for short. So a SNP is a variation in nucleotide at a specific position in the genome. And we're going to be interested here in non-Gaussian phenotypes, such as counts and binary phenotypes. So for an example of counts would be number of root-like structures in a plant. And an example of a binary phenotype would be, does the subject have or do not have the disease? And then for these non-Gaussian phenotypes, we're going to consider generalized linear mixed models. One of the issues in GWAS analysis is that different individuals of a species will have correlated responses because of similar population structure. And to take into account such correlation among the observations, usually statistical analysis of GWAS data includes random effects with a covariance matrix that's proportional to a kinship matrix that's assumed to be known. And in population studies, this kinship matrix is estimated from SNP data. In order to identify SNPs related to phenotypes, here we propose a Bayesian analysis of GLMMs for GWAS data. Essentially, this is a model selection problem for GLMMs where P, the number of SNPs, is way much larger than the number of observations N. The observations are correlated, the SNPs regressors are highly correlated. If you try to fit this using MCMC, that's going to be unfeasible. And then if you try to do something like stochastic model search on the original model space, that will most likely not find the best model. Here I propose BG2, Bayesian GLMMs for GWAS. Some key features of BG2 are the following. We use a pseudo-likelihood approach to help integrate out random effects. BG2 is a two-step procedure with a screening step and a model search step. We use an approximation that's called population parameters previously determined. And we also develop novel non-local priors for the regression coefficients of the SNP regressors. Generalized linear mixed models for GWAS. So here is a GLMM for GWAS. We have a PDF for yi, which is in the exponential family of distributions. So it's equal to exp of yi theta I minus bi theta I plus ci yi. And then we have a link function g that relates theta I to a linear predictor that's written as xi transpose beta plus zi1 transpose alpha 1 plus zi2 transpose alpha 2. The functions bi and ci are known, and they depend on which distribution yi follows. So if the Poisson distribution, then bi and ci are going to be certain functions. If it's the Bernoulli distribution, bi and ci are going to be different functions. If it's the gamma distribution, bi and ci will be other different functions. But all of those functions are known if we know what's the distribution of yi. Here we assume that y1, y2, etc., yn are conditionally independent phenotype responses. Xi is a vector that has SNP regressors and other control covariates, such as, for example, sex and age. Beta is a vector of regression coefficients. Alpha I is a vector of kinship random effects. Zi1 is an incidence vector of these kinship random effects for the ith observation. Alpha 2 is a vector of overexpression random effects, and Zi2 is an incidence vector of overexpression random effects. Here, even though the yi's are conditionally independent, they become correlated once we take into account the kinship random effects. So if we integrate out the kinship random effects, that induces correlation among the yi's. The model for the kinship random effects is a multivariate Gaussian distribution with a mean vector of zeros and a covariance matrix that's an unknown variance parameter kappa 1 times a non-kinship matrix sigma 1. And the model for the over-dispersion random effects is a multivariate Gaussian distribution with a mean vector of zeros and a covariance matrix that's just a unknown variance parameter kappa 2 times the identity matrix. So we're assuming that the elements of this vector alpha 2 are independent. Let me tell you about the integrated likelihood. So the integrated likelihood obtained by integrating out the kinship and over dispersion random effects is given by this expression here. So it's a multidimensional integral, and unless the yi's are Gaussian, so for other cases, this integral cannot be computed analytically, and we cannot use MCMC in this problem because we have 200,000, 800,000, or sometimes over 1 million SNPs to deal with. So we approximate the exponential family likelihood function with an approximate Gaussian likelihood function. And for that we use a pseudo-likelihood approach. This then provides an approximate Gaussian linear mixed model for what's called adjusted observations Y star, and then the random effects can be analytically integrated out of this approximate Gaussian model. And we should note that John et al. In 2021 have used a pseudo-likelihood approach to develop a frequentist single marker analysis for GWAS binary data, non-local prior. As shown by Johnson and Russell, non-local priors allow faster accumulation of evidence in favor of simpler models when compared to local priors. And Johnson and Russell proposed non-local priors for Gaussian linear models. And Wu et al. In 2020 proposed non-local priors for generalized linear models. But to the best of our knowledge, the prior we propose here is the first non-local prior for generalized linear mixed models. So here is the prior that we propose for beta. The kernel is a Gaussian kernel times a product of the elements of the beta vector squared. So what this is going to do is when any element of the beta vector gets close to 0, this prior density is going to get close to 0. Here, this matrix H is the approximate covariance matrix of the adjusted observations after integrating out the random effects. So here we have X transpose H to minus 1X divided by N as the precision matrix of the prior for the beta vector before we multiply by the square of each element of the beta vector. Just to have some visualization of the difference between non-local priors and local priors. So the dashed line here is a usual Gaussian local prior. And then we have two non-local priors. The dotted line here represents a non-local prior with parameter tau equal to 0.348. And the solid line represents a non-local prior with parameter tau equal to 0.022. SNP search. The most used GWAS procedure is called single marker analysis, or SMA for short. And what SMA does is it fits as many models as the number of SNPs, where each of those models considers only one SNP. Then each of these fitted models has an associated p-value, and then SMA applies either Bonferroni correction or Benjamini-Hochberg FDR correction to correct for multiple tests. And then SNPs with small corrected p-values are reported. Now, what happens here is that the SNPs are highly correlated. So if you have a SNP that's causal, that has a strong effect size, then all of those SNPs that are highly correlated to that one SNP also show up as important SNPs, but they're not. And then that leads to a high FDR. In contrast, our proposed Bayesian approach has two steps. First, a screening step that fits as many models as the number of SNPs, where each model considers only one SNP. Then we use Bayesian FDR control, as proposed by Newton et al. And Miller et al., to select a set of candidate SNPs. And then we use a model search step that performs a stochastic search through model space with a genetic algorithm. Each model here is a GLMM that may include multiple SNPs from the candidate set obtained from the screening step. And then BG2 reports the SNPs in the model with the largest posterior probability. And we are going to show later with our simulation studies that this model search step reduces the number of false positives tremendously. Let me now talk a little bit about this approximation, the population parameters previously determined approximation. When we're dealing with linear mixed models, an expensive part of the computations is the numerical optimization to estimate the variance parameters. And then when performing SMA with LMMs, then computing estimates such as MLEs and REMOs can be very costly from a computational point of view. And of course, for GLMMs, this is even more costly. One solution is an approximation called population parameters previously determined, or P3D, that was first proposed by Zhang et al. In 2010 for GWAS analysis using linear mixed models. So for GWAS LMMs, the P3D approach of Zhang et al. Estimates the variance parameters fit in an LMM without any SNPs, and then uses these estimates as the true variance parameters while performing SMA. And then there's a variation of this approximation that's used in the Emacs method that makes that method very fast. And this P3D approximation works well for GWAS data because the effect sizes are usually small for each SNP. Now here we also propose a P3D approach that's somehow different from those P3D approaches from the previous slide. So how does our P3D approach work? At each step of SNP search, we estimate the variance parameters fit in a baseline model. And then this model fit also yields adjusted observations. We then use this variance parameter estimates and the adjusted observations while we fit all other models in the SNP search step. So while we fit all other models in the SNP search step, we do not update the variance parameter estimates and we do not update the adjusted observations. So this saves a lot of computations. In the screening step, the baseline model is a GLMM without any SNPs. And in the model search step, the baseline model is a GLMM with all candidate SNPs. Screening and model selection details. First, let's talk about the screening step details. So we fit the baseline model with no SNPs. And with that, we obtain adjusted observations Y star and estimates for the variance parameters. Now let H be the approximate covariance matrix of the adjusted observations Y star according to the baseline GLMM after we integrate out the random effects. And because we are fixing the variance parameters at their estimates based on the baseline GLMM, H is known. And now we fit as many approximate LMMs as the number of SNPs to the adjusted observations. Now the model fit for, say, SNP S yields a maximum pseudo likelihood estimator beta S hat for the regression coefficient beta S. The distribution of beta S hat given beta S is approximately a Gaussian distribution with mean beta S and with variance sigma S squared, where sigma S squared is just the reciprocal of XS transpose H2 minus 1 XS. Now, in the screening step, we assume that beta 1, etc., beta P are a priori exchangeable, and we assume that each of them follows a prior that is a mixture of a point of mass at 0 and our proposed non-local prior, specialized for the case of a scalar beta S. So here is the prior PDF. Pi 0 is the probability of the null hypothesis that beta S is equal to 0, and tau larger than 0 is a scale parameter. And then we estimate pi 0 and tau by maximizing an approximate log likelihood function, which is the summation for S from 1 to P of the log of the PDF of beta S hat given tau and pi 0. And this PDF can be obtained as an integral. We can compute this integral analytically. The advantage of being able to compute this integral analytically is that then our implemented procedure is very fast. Then what we do is we take an empirical based approach. We keep pi 0 and tau fixed at their estimates, and then we apply Bayes' theorem to compute the posterior probability of beta S being different than 0 for S being 1 through P. And then we use Bayesian false discovery control to keep the false discovery rates nominally at 5%. And what this screening step does is it provides a set of candidate SNPs for the next step of SNP search. Now let's talk about this next step. The next step is model search. So say that we have P1 candidate SNPs from the screening step. Then the model search step fits the baseline GLMM with all those P1 candidate SNPs, obtaining estimates for the variance parameters and adjusted observations Y star. Now let H be the approximate covariance matrix of the adjusted observations according to this baseline GLMM. So note that the baseline GLMM in the model search step is different from the baseline GLMM from the screening step. The baseline GLMM in the model search step includes all P1 candidate SNPs that were obtained from the screening step. Now consider model MK. K here varies from 1 to 2 to the P1. Let MK be the number of SNPs in model MK. Then in the model search step, we assume that the prior probability of model MK is this prior probability here. Here, pi 0 hat the estimate of pi 0 from the screening step. And then we use for beta k this non-local prior that we talked about previously. Here, dk is a normalizing constant. In the paper, we provide details about how to compute that normalizing constant. And then if P1 is small, that is P1 is like less or equal than 16, then BG2 uses Bayes' theorem to compute the posterior probabilities for all the possible models. However, if P1 is large, then we cannot do this exhaustive enumeration of the models. So if P1 is large, then BG2 uses a genetic algorithm that searches through model space. And finally, in both cases, BG2 reports the SNPs that appear in the largest posterior probability model. Simulation studies. We have two simulation studies. The first one has binary data and considers human SNPs. The second simulation study is with count data and considers Italian SNPs. Here we compare BG2 to SMA with Bonferroni correction. We have 4 criteria: number of true positives, number of false positives, false discovery rates, and F1 score. To compute this criteria, we simulated 100 data sets under each simulation condition, and then we average those criteria over the 100 simulated data sets. Now, we use a buffer concept to decide what is a true positive and a false positive. Specifically, if one or more SNPs adjacent to a causal SNP are detected, that is counted as a true positive. And then each detected SNP that's not adjacent to a causal SNP is counted as a false positive. And we consider BG2 here with 4 different ways of dealing with the non-local prior scale parameter tile: a uniform prior, an inverse gamma prior, which has mean 0.022. We also consider fixing tau at 0.022 as proposed by Sanyal et al. In 2019, and fixing tau at 0.348 as proposed by Johnson-Russell in 2010. Okay, so the first simulation study to binary data, we consider 800,000 SNPs with minimum allele frequency larger than 0.01 from 2,772 European Americans from the study of addiction, genetics, and environment. We assume 20 causal SNPs, 10 SNPs with small fixed coefficients with absolute values equal to 0.2. And then the other 10 SNPs have the same regression coefficient, which we vary here from 0.2 to 1, so that we can see what happens as the effect size increases. We have a kinship random effect, alpha 1 vector. The kinship variance parameter is equal to 0.15 here. And we simulate data from this binary GLMM. Intercept is minus 1.5 here. Here are some plots that show the results. The top left plot shows the true positives as a function of the value of beta. Here, the dashed blue line shows the results for the inverse gamma prior. The light solid blue line shows the results for the uniform prior. The solid gray line shows the results for fixing tau equal to 0.022. And the solid black line shows the results for SMA. So in the true positives, as beta increases, SMA actually is able to find more true positives than our methods. The problem is that what happens with the false positives? SMA has a much larger number of false positives. And the number of false positives actually increases as beta increases, whereas our methods control the false positives very, very well. In terms of the false discovery rates, SMA has a very large false discovery rate, whereas our methods control the false discovery rate in a much better manner. And then in terms of F1 score, larger is better, so you can see that our method with the uniform prior is better than SMA for any of the considered values of beta. Let's take a look at another simulation study. So this one is with count data. We have a little less than 200,000 SNPs with minimum allele frequency larger than 0.01. And these come from 152 ecotypes of Italiana. And here we are going to assume that each ecotype has 12 replicates, so 12 biological replicates. We have 10 causal SNPs, 5 SNPs with small fixed coefficients, beta j equal to 0.4, and then other 5 SNPs that have the same regression coefficients, which vary here from 0.1 to 1. Here the kinship random effect is going to have a variance parameter equal to 0.1, and the over-dispersion random effect is going to have a variance parameter equal to 0.3. These are inspired by our analysis of real data for this species. And then the data are simulated by this Poisson GLMM. Here are the results. Again, the color coding is the same as previously. In terms of true positives, it's interesting that our methods actually have increased the number of true positives as beta increases, whereas SMA suffers in that regard. In terms of false positives, the methods are pretty comparable up to when beta is about 0.5-ish, but then after that SMA starts getting lots and lots of false positives. And then in terms of the false discovery rates, things are pretty similar up to beta equal to 0.5, but then SMA starts getting much larger false discovery rate as beta increases. In terms of F1 score, for small values of beta, these methods are all performing very similarly, but as beta increases, our methods perform much better than SMA. Let me present a couple of applications. The first application is about maximum number of alcoholic drinks. The data are from the collaborative study on the genetics of alcoholism. The phenotype is maximum number of alcoholic drinks consumed in 24 hours. To analyze this data set, so this is count data. So we use the Poisson GLMM with kinship random effects and over-dispersion random effects. The genetic covariates are 846,076 SNPs with minimum allele frequency larger than 0.01 and with less than 5% missing from 2,759 European Americans. While SMA detects 10 SNPs, BG2 detects only 1 SNP. To give more specific information, the screening step of BG2 identified 10 candidate SNPs. Those 10 candidate SNPs were then given to BG2 model selection step. And then in the model selection step, BG2 identified just 1 SNP. This SNP is located in the protein coding gene PTGER4 on chromosome 5. The protein encoded by this gene is a receptor of prostaglandin E2, and an increase in PGE2 is part of the inflammatory response to alcohol consumption. And the use of an inhibitor of PGE2 significantly reduces the severity of several hangover symptoms. So it's interesting that this seems to be an important gene that BG2 has found. Another application is on human cocaine dependence. The data are from the family study of cocaine dependence. That's a part of the study of addiction, genetics, and environment. The phenotype is DSM-4 cocaine dependence, so it's binary data. So for each subject, it says, does this person have cocaine dependence or not? And to analyze this data set, we use the Bernoulli GLMM with kinship random effects. Genetic covariates are 846,076 SNPs with math larger than 0.01 and with less than 5% missing from 2,767 European Americans. And we use sex as a control covariate because males and females seem to have very different proportions of cocaine dependence. Our method BG2 detects one SNP. SMA also selects the same SNP. And this is located in the protein coding gene ABCC8 on chromosome 11. And this gene is a member of a subfamily of genes involved in multi-drug resistance. And an interesting piece of information is that recent experiments with mice have shown that another gene in this subfamily, ABCC1, has increased expression with cocaine use. And in addition, if a silencer of ABCC1 is given to the mice, that blocks cocaine-induced place preference. So it seems that this is another important gene to consider. Here's another application. This is to root-like structures in A. Italiana, so it's a plant science application. Data are from an experiment with A. Italiana explants. The phenotype is the number of root-like structures, so this is count data. Because of that, to analyze this data set, we use the Poisson GLMM with kinship random effects and over-dispersion random effects. The genetic covariates are a little less than 200,000 SNPs, with MAF larger than 0.01 from 152 ecotypes, with 12 biological replicates per ecotype. BG2 detects 3 SNPs, which are expressed in the root and are located in protein coding genes AT1G20090, AT1G20100, and AT1G20720, and SMA detects the same SNPs. It's worth to note that AT1G20100 encodes a DNA ligase-like protein involved in the regulation of metabolic processes. In addition, AT1G20720 encodes a RAD3-like DNA-binding helicase protein that acts in the repair of double-strand breaks in DNA and also in nucleotide excision repair. And finally, AT1G20090 encodes a ROP2 protein, which is known to affect root hair initiation and tip growth. So these 3 genes seem to be related to root-like structures in A. Italiana. Conclusions. So just to wrap up, we propose BG2, that stands for Bayesian GLMMs for GWAS. BG2 is a 2-step Bayesian SNP search for GWAS data in the exponential family. So this is able to analyze binary data and count data, for example. We propose a non-local prior for the coefficients of the covariates, and simulation studies show that when compared to SMA, BG2 has a much lower FDR. Now, we must say that there are some combinations of effect sizes for which SMA and BG2 provide similar results. And that is what seemed to have happened in the applications on cocaine dependence and number of root-like structures. However, in some applications, BG2 provides a much smaller number of false discoveries than SMA. And that's what seems to have happened in the case study on maximum number of alcoholic drinks. So when we compare BG2 to SMA, BG2 is more robust and precise. Thank you. If you have any questions, please send me an email at marf@vt.edu. Thank you.